All right, so how the nerve impulse actually works. Basically, the sodium-potassium pump is maintaining a situation where inside the cell is more negative than outside the cell. So what we see happen inside, it's approximately negative 70 millivolts. It's a measure of voltage. When the nerve gets stimulated, what will happen is there's these gated channels, sodium-gated channels. We're going to look at how this works in a second. They open. And when the gates open, the sodium that's accumulated outside quickly flows in and temporarily makes the inside positive. And that's called depolarization. If this is a strong enough signal, it causes what's called an action potential, which basically flows down the nerve. It basically stimulates, if we imagine there's a channel, this one is open, it stimulates the one next to it to open, which stimulates the one next to that to open, and this impulse travels down the neuron. As soon as this channel opens, the gate slowly closes again. And when that happens, it starts to restore this section back to normal, and the impulse is again moving to the next section. One of the things that helps to restore it back to normal are potassium-gated channels that open, that start moving potassium out, and so the positive charges are being moved back outside. The sodium-potassium pump helps everything to reset. There is a small moment of time where that nerve cannot be stimulated again until everything is reset. So, for example, if you were to smack a table really hard right afterwards, your hand might feel numb. You wouldn't actually be able to stimulate that nerve again until it's reset. So if we look at resting potential, on the right side here we have pictures of this. We have, this is resting potential in our first picture. Here the nerve gets stimulated, and this is supposed to be the sodium-gated channels opening and sodium rushing in. And that correlates to number 2 here on the graph. Sodium's rushing in, it becomes positive. It peaks here at the top. Now the potassium channels open, and we see here the gated potassium channels open in our third one, and this starts to restore it back to normal. There's a little bit of an overshoot here. This is actually, we're not going to talk about it. This is actually called hyperpolarization, where the nerve becomes hyperpolarized, and then it's back to normal. Notice when the nerve is stimulated, you can have a very small stimulation of some dendrites here on the end. If only a couple little channels open, and it may not actually cause an action potential. There's this number right here, this negative 55. This is what's called the threshold. So every single thing that touches your dendrites doesn't necessarily stimulate a neuron, or your neurons would be stimulated all the time. So there's sort of a minimum that's needed, sort of like dropping a little pebble in a pond might make a couple ripples, but it's not enough to go all the way across the pond. And that's what's happening here. So you need enough of a stimulus that it brings that from negative 70 to negative 55, and if that happens, that's what will cause the nerve to depolarize and cause that chain reaction. Remember, when it gets to the end, that's where the neurotransmitters are that would stimulate the brain, the spinal cord, the next neuron, the muscle, whatever it might be. But the neuron's not actually attached to that. It just releases chemicals that would then stimulate the next one in the process. The last kind of signaling is called long-distance signaling, because rather than just stimulating the cell next to it or the cells in the local area, hormones can carry a signal through your endocrine system from a cell that's, let's say, in your brain, in your pituitary gland, all the way to cells that are, in the ovaries, or your thyroid, or your adrenal glands. So there's glands all over your body that are releasing hormones that then travel to target cells that are located far away. So the hormones have to travel through the bloodstream. Note that plants also have hormones. They don't have a bloodstream, but for example, they can release hormones as gases, like ethylene, for example. Ethylene is a plant hormone that causes fruit to ripen. So if you take a very ripe banana and you put it with a bunch of other bananas, it'll actually make the other bananas start to ripen. And farmers can actually spray ethylene on plants to make the fruits ripen faster so they can get the fruits to market. So hormones, all you really need to know from this slide is that long-distance signaling uses hormones. They are made in one area and then carried through the circulatory system to another area where they reach what are called their target cells. You know, hormones don't actively look for the cells they're going to stimulate. Hormones pretty much flow through your blood and hit every cell, but only cells with receptors to that hormone would actually respond to it. And a lot of times you have hormones that work together. So, for example, insulin lowers blood sugar, but if the blood sugar gets too low, glucagon can raise blood sugar. You also have calcitonin and parathyroid hormone, which work together to raise and lower the levels of calcium in your blood. And other hormones work the same way. Estrogen and progesterone work together. So there's different hormones in your body that work together to keep everything stable, bring things up, bring things down, and maintain stability. Here's a picture showing it. So the hormone's made in one area, it travels all the way to another area. There are 3 steps in the way hormones work. One is reception. They have to attach to a receptor. The second is transduction, and this is where the signal will get processed sort of inside the cell and amplified. And then the response is whatever that cell does in response to the hormone, like opening a gate, making a protein, whatever it might do. Here's sort of a visual of it. Reception at the cell membrane in this particular picture. Relay molecules, that's the transduction. And then the response is some kind of an activation. 2 different kinds of hormone receptors. Water-soluble hormones, which in general, we're going to keep it simple, are your protein hormones. They're going to bind to a receptor on the membrane. In other words, a protein hormone cannot actually go into the cell. So it'll bind to a receptor here, and then the response will happen. Lipid hormones, which would basically be your steroid and thyroid hormones, they can go right through the cell membrane, through the phospholipid bilayer. So if this is my hormone, and the receptor is usually something that's inside the cell. It could either be in the cytoplasm or even in the nucleus, but it would bind there and then cause some kind of a response. So that's the difference. They all have receptors that have to pick up the signal. Without a receptor, the cell's not going to respond. But protein hormones bind to a receptor on the surface, to a protein receptor on the surface of the cell membrane. Lipid hormones, which are pretty much steroids, that's the one to really remember, they go right through the cell membrane, and they still have to find a receptor, but the receptor will be inside of the cell. So this is what a steroid hormone would look like. It would go straight through the cell membrane. Notice it finds its receptor. The receptor goes to the nucleus, and in this case, this hormone is causing a protein to be made. It's showing transcription, some stuff we haven't talked about yet, but in essence, it's turning on the DNA to make some kind of a protein. So that would be a steroid. Once the receptor is stimulated, the second step, transduction, amplifies the signal. In other words, you could have one hormone bind to the surface, and then what happens inside is something that maybe causes 100,000 molecules to be made through a series of steps where this one binds to this one, which binds to this one. And so you don't need, it's not a one-to-one thing, whereas one nerve stimulates one thing next to it, one hormone can cause a massive response. And this can control how much of a response you get, depending on how much hormone you make. The relay molecules inside the cell are usually proteins, and they, like I said, what they do, they are sort of like a chain reaction. Each step sort of activates another step in the pathway until you get your final change. So one changes another. I'll show you, here's a kind of a picture of what it looks like. So you have your signal on the outside. This causes this protein to get activated, which then stimulates a second protein to get activated, which then causes a third protein to get activated. And one hormone might cause 100 of these to happen, which might stimulate down here 100 of each of those 100 of these. So you can end up with 10,000 or 100,000 or even 1 million product molecules as a result of this chain reaction from one hormone binding. And again, the response is either maybe making an enzyme, turning on or off a particular segment of DNA, a gene, to make a particular protein, maybe opening an ion channel, activating something that was inactive. There's all kinds of things that can happen as a response. Here's again another thing. So you have one hormone binds, a whole domino effect inside, and at the end, some kind of a protein is being made. So, and that's just the summary of the same thing. So this is a YouTube video that somebody made for an AP biology class. This is showing a hormone that stimulates glycogen to be broken down into glucose. So there's the hormone binding. And again, you don't have to know the details. The hormone is epinephrine in this case. Epinephrine binds, and now this is what's going to start this chain reaction. So basically one hormone and 100 of these G proteins are activated. Again, you don't necessarily have to understand that. This is going to cause a chain reaction, which you're going to see now. This makes ATP, which then makes something called cyclic AMP, and this is going to stimulate these proteins in this pathway. Again, one hormone is actually causing 100 of these. So they just drew one of these going through this chain reaction, but there technically would be like 100 of these. It finally goes to glycogen, and now glycogen starts to get broken down. And in essence, you started with one epinephrine, and finally, how many glucose 1-phosphates are made from the breakdown? I believe that was like 100 million. So that kind of gives you an idea of what's happening.